Right now, you are breathing. You are not thinking about it, you didn't plan it, it's just happening. But if something were to slice through your spinal cord right at the base of your neck, specifically above the C3 segment, that automatic rhythm vanishes instantly. Your body completely forgets how to breathe. Why? Because breathing isn't generated by your lungs. It's driven by a precise rhythmic network of neurons buried deep in your brainstem. Today, we are going inside the master clock of respiration to see how your brain controls every breath, how it senses chemical shifts in your blood, and what happens when the entire system is hijacked. Meet the literal pacemaker of your breath, the pre-Porzinger complex. Tucked inside the medulla, this tiny network of synaptically coupled pacemaker cells fires continuous electrical bursts to establish your baseline respiratory rate. But these cells have a major vulnerability. They are packed with opioid receptors. This is exactly why opioids are so dangerous. When a patient takes high doses of opioids for pain, the drug binds to these opioid receptors, shutting down the pre-Porzinger complex. This triggers potentially fatal respiratory depression. The brain literally forgets to tell the body to inhale. The pre-Porzinger complex doesn't work alone. It coordinates a team of two primary neuronal groups in the medulla. The dorsal respiratory group, found inside the nucleus of solitary tract. This zone is all about inspiration. These neurons fire down the spinal cord to command the motor neurons of your phrenic nerve, causing your diaphragm to contract and draw in. And the ventral respiratory group, running through the nucleus ambiguous. This group handles both inspiration and forced expiration. During restful breathing, the ventral respiratory group stays quiet, but the moment you exercise or force an exhalation, it wakes up to drive your internal intercostal and abdominal muscles. To keep things clean, the system uses a strict pattern of reciprocal innervation. When inspiratory neurons fire, expiratory neurons are completely paralyzed, and vice versa. This raw medullary rhythm is constantly modulated by higher ranking centers in the pons. The pneumotaxic center located in the upper pons is the internal off switch for inhalation. It continuously signals down to the medulla to limit how long an inhalation lasts, which naturally speeds up your breathing rate. If you damage this center, your breaths become profoundly deep and slow. The apneustic center, located in the lower pons, wants the exact opposite. It fights to prolong inhalation. If a scientist surgically destroys the upper pneumotaxic center and simultaneously severs both vagus nerves, the brain enters a bizarre state called apneusis. The subject experiences prolonged, agonizing inspiratory spasms, effectively getting stuck at full inhalation. This brings us to Herring-Brewer reflexes, which travel up the vagus nerve to prevent mechanical damage. As your lungs expand, slowly adapting stretch receptors in your airway, smooth muscles start firing. Once that signal hits a critical threshold, it cuts off the inspiratory neurons, acting as a hard safety ceiling to keep you from overinflating and tearing lung tissue. Massive deflation triggers a reflex which prematurely cuts exhalation short to force an immediate protective rescue breath. We actually see this play out in heart-lung transplant patients. Because the donor lungs are severed from the recipient's vagal pathways, these patients completely lack the Herring-Brewer reflex, yet they breathe perfectly normally at rest. This proves that in humans, the Herring-Brewer reflex is a protective emergency backup, not a daily regulator of resting breath. To keep your blood chemistry perfect, your brainstem relies on two distinct families of sensors: central and peripheral chemoreceptors. Central chemoreceptors are located on the surface of the medulla. These sensors are completely blind to oxygen. They also cannot detect hydrogen ions in your blood because the tight blood-brain barrier blocks charged particles from crossing. Instead, they track carbon dioxide. Because CO2 is a nonpolar gas, it diffuses across the blood-brain barrier effortlessly. Once inside the CSF, it reacts with water to form carbonic acid, which breaks down and releases free hydrogen ions. Since the CSF has almost no protein buffers, the pH plummets. This ramps up ventilation and blows off excess CO2. Peripheral chemoreceptors live outside the brain in carotid and aortic bodies. The carotid body is an absolute engineering marvel, relying on specialized type 1 glomer cells to read oxygen levels. Under normal conditions, unique oxygen-sensitive potassium channels are wide open, letting potassium leak out freely. The moment arterial pressure of oxygen drops below 60 millimeters of mercury, these potassium channels slam shut. Potassium builds up inside, driving the cell's voltage upwards, causing depolarization. This electrical shift opens voltage-gated calcium channels. Calcium rushes in, pushing dense core granules to release neurotransmitters. These neurotransmitters cause action potentials to travel up the glossopharyngeal nerve into the brainstem, triggering emergency hyperventilation. The carotid body has a massive blood flow relative to its tiny size, an unbelievable 2000 ml per 100 grams of tissue per minute. Because this flow is so astronomical, its metabolic needs are met entirely by physically dissolved plasma oxygen alone. This means the carotid body is completely blind to anemia or carbon monoxide poisoning. In these conditions, hemoglobin oxygen drops drastically, but the dissolved arterial partial pressure of oxygen stays normal at 100 millimeters of mercury. Because dissolved pressure doesn't drop, the glomerulus cells never depolarize and the body never triggers an emergency breath response. These regulatory feedback loops are very important for life. And when these cross paths with trauma, things get dangerous fast. Let's discuss about drowning. In 10% of cases, the first drop of water hitting the larynx triggers a massive laryngospasm. The airway clamps shut, causing death by drowning without a single drop of water entering the lungs. In the other 90%, fluid floods the tissue, creating radically different pathology based on salinity. In freshwater drowning, hypotonic fluid is vacuumed across into the bloodstream, diluting plasma and violently rupturing the red blood cells. In saltwater drowning, hypertonic ocean water acts like a sponge, pulling fluid out of your blood vessels into the air spaces, causing severe pulmonary edema. Finally, there's obstructive sleep apnea. During sleep, there is loss of muscle tone and the muscles can no longer pull the tongue forward. The tongue collapses backward, completely sealing off the airway. The patient fights against a closed pipe, waking up briefly to catch their breath hundreds of times a night. This chronic, fragmented sleep and oxygen starvation multiplies their risk for car accidents sevenfold and triggers long-term sympathetic damage leading to hypertension, stroke, and right heart failure. From the microsecond pacing of pre-Porzinger complex to elegant potassium channels of the carotid body, the respiratory control system is an incredibly fine-tuned balance of chemistry, mechanics, and physics. Next time you take a deep breath, remember the complex machinery working behind the scenes to keep that air moving. Hit that like button if you uncovered something new today. Subscribe to the channel for more deep architectural dives into the human body, and I will see you in the next one.